Dr. Zihao Ou, assistant professor of physics at the University of Texas at Dallas, and his team have unlocked a new frontier in biomedical research with their groundbreaking technique to render live mouse skin transparent using tartrazine, a yellow dye commonly found in snacks and candies. This innovation, unveiled in the September 6th issue of the journal Science, paves the way for unprecedented insights into live biological processes and disease mechanisms. By applying a mixture of water and tartrazine to the skin on the skulls and abdomens of live mice, the researchers succeeded in making the skin transparent. This remarkable transparency was achieved through the unique interaction between the dye and the skin tissue. Living skin functions as a scattering medium that, much like fog, diffuses light and prevents it from passing through. Tartrazine, on the other hand, is a light-absorbing molecule that primarily absorbs blue and ultraviolet light. When these two elements, skin and the dye, were combined, the team found that they could cancel out each other's light-blocking properties, allowing light to pass through and make the skin appear transparent. This technique was developed during Ou's tenure as a postdoctoral researcher at Stanford University before he joined UT Dallas. We combined the yellow dye, which absorbs most light, especially blue and ultraviolet light, with skin, which scatters light. Individually, these two things block most light from getting through them. But when we put them together, we were able to achieve transparency of the mouse skin, O explained. The implications of this study are far-reaching, offering the potential for non-invasive imaging techniques, deeper understanding of skin-related diseases, and more effective drug delivery methods. As O and his team continue to refine this technique, the possibilities for its application in medical science and research are both exciting and vast. In their sterile laboratory, a group of researchers gathered around a small lab bench, staring intently at a curious sight: a small mouse with an almost eerie transparency to parts of its body. Dr. O, the lead scientist, gently picked up the creature, turning it over to give his colleagues a better view. He pointed to its abdomen, where the small intestine's rhythmic contractions were easily visible through the now transparent skin. For those who understand the fundamental physics behind this, O began, it makes sense, but if you aren't familiar with it, it looks like a magic trick. His colleagues nodded, murmuring among themselves about the implications. The magic happened because dissolving light-absorbing molecules in water altered the solution's refractive index in a way that matched the tissue component's refractive index, such as lipids. Essentially, these dye molecules reduce the degree to which light scattered in the mouse's skin tissue, much like how dissipating a fog bank reveals the scenery behind it. Their successful experiment was not just a scientific curiosity, it had broad practical applications. O recounted how they rubbed the dye solution onto the skin of mice, focusing on their skulls and abdomens. Once the dye completely diffused into the skin, it became transparent, O explained. The transformation took mere minutes, similar to how a facial cream or mask works, requiring time for molecules to diffuse into the skin. Observing through the transparent skin of the skull, the researchers had directly seen blood vessels on the brain's surface. In the abdomen, they watched internal organs and peristalsis, the muscle contractions responsible for moving contents through the digestive tract, in real time. It's reversible too, O added, just wash off any remaining dye, and the dye that is already diffused is metabolized and excreted through urine. Although the transparent areas of the mice had taken on an orangish hue, there was a significant advantage to the dye they used: FDNC yellow number 5, or tartrazine. Commonly found in snack chips, candy coatings, and other foods, it was biocompatible, inexpensive, and efficient. The dye is certified by the FDA and safe for living organisms, O affirmed. The conversation then turned toward the human applications of their breakthrough technology. O mentioned that human skin, being roughly 10 times thicker than that of a mouse, posed a significant challenge. They had not tested the dye on humans, and it was unclear what dosage or method of delivery would be required to penetrate the thicker skin. In human medicine, we currently have ultrasound to look deeper inside the living body, Ou said. Many medical diagnostic platforms are very expensive and inaccessible to a broad audience, but platforms based on our tech should not be. The room buzzed with excitement and cautious optimism about the future possibilities, as the researchers pondered how this seemingly magical trick could revolutionize medical diagnostics. Dr. Zihao Ou earned a Bachelor of Science in Physics from the University of Science and Technology of China and a doctorate in Materials Science and Engineering from the University of Illinois Urbana-Champaign. His doctoral research focused on electron microscopy, an imaging technology that uses beams of electrons, instead of light, to produce high-resolution, magnified images of biological and non-biological specimens. I know my field very well, but after I finished my PhD, I wanted to do something that would have more of an impact on more people, rather than limit myself to materials science, Ou said. So I decided to learn some biological imaging techniques and bring my background in physics and materials science to biomedical science. As a postdoc, I believe my background in physical science brought a unique perspective to our biological imaging research. In his new dynamic bioimaging lab at UTD, Ou will continue the research he started with Dr. Guosong Hong, assistant professor of materials science and engineering at Stanford and a corresponding author of the study. Ou said the next steps in the research will include understanding what dosage of the dye molecule might work best in human tissue. In addition, the researchers are experimenting with other molecules, including engineered materials that could perform more efficiently than tartrazine. Study authors from Stanford, including co-corresponding author Dr. Mark Brongersma, the Stephen Harris Professor in the Department of Materials Science and Engineering, were funded by grants from federal agencies including the National Institutes of Health, the National Science Foundation, and the Air Force Office of Scientific Research. As an interdisciplinary postdoctoral scholar, Ou was supported by the Wu Tsai Neuroscience Institute at Stanford. The researchers have applied for a patent on the technology. Ou said one of the first applications of the technique will likely be to improve existing research methods in optical imaging. Our research group is mostly academics, so one of the first things we thought of when we saw the results of our experiments was how this might improve biomedical research, he said. Optical equipment, like the microscope, is not directly used to study live humans or animals because light can't go through living tissue. But now that we can make tissue transparent, it will allow us to look at more detailed dynamics.